This video is used to introduce the main sequencing methods currently available. They are Sanger sequencing and next-generation sequencing. In 1977, Frederick Sanger invented Sanger sequencing, also known as the chain termination method. He became a two-time Nobel laureate in this way. First, we will introduce the mechanism of Sanger sequencing. We know that the four bases in DNA are adenine, cytosine, guanine, and thymine. These bases form specific pairings A with T and G with C. DDNTP refers to dideoxide nucleotides triphosphates. It lacks a 3-hydroxide group compared to DNTPs. DDNTPs are chain extension inhibitors of DNA polymerase. We will prepare 4 different DDNTP. First, these are our DNA. We raise the temperature and the DNA will become single-stranded. The DNA is then annealed to bind to the target primer, and it begins to extend in the presence of DNA polymerase. DDNTP was added continuously during the elongation process to stop the chain extension. As a result, several chains of different lengths were formed, and they all end with the same DDNTP. We will repeat the same experiment 4 times so we can get all the different lengths of chains ending with the 4 DDNTPs. Next, we ran the products from the 4 tubes through electrophoresis. We can observe that each band has its corresponding position. The shorter chain run at the front, while the longer ones run more slowly. Then we read the sequence in reverse order. In 1987, the automated Sanger sequencing was invented. The 4 DDNTPs were labeled with 4 different fluorescence. The main difference between automated Sanger sequencing and the previous one is the use of laser and the detector for automatic sequencing. As the differently labeled capillary gels pass through the laser and detector, fluorescence will identify each DDNTP. The laser excites the fluorescent marker in each band, and the detector produces the resulting emitted light. Because each of the 4 DDNTPs is labeled with a different fluorescent marker, the emitted light can be directly correlated with the identity of the terminal DDNTP. The computer outputs a chromatogram which shows a fluorescence peak for each nucleotide along the length of the template DNA. The Sanger sequencing method has the advantages of obvious visualization, simple operation, and low false positive potential. At the same time, it becomes indistinguishable when there is a stream of same bases in the target sequence. In addition, it has the disadvantages of low flux and time-consuming.
Sequencing technology has continued to evolve over the last few decades. Second-generation sequencing systems have been introduced within the last decade to meet the requirements of large-scale sequencing. Those include, but are not limited to, Illumina MiSeq, Illumina HiSeq, 454 sequencer, soiled system. Those systems are capable of analyzing millions or even billions of sequencing reactions simultaneously. Here are some of the machines with different technical details about NGAs. However, they have some common features in existence. First one, sample preparation. All next-generation sequencing platforms require access to a library linked by amplification or custom adapter sequences. 2. Sequencing machines. Each library fragment is covalently amplified on the soiled surface by negating DNA linkers. It hybridized to the library adapter. This amplification produces DNA clusters, each from a single library fragment. Each DNA cluster will be used as a separate sequencing reaction. 3. Data output. Each machine provides raw data at the end of the sequencing run. This raw data is the collection of DNA sequences generated for each DNA cluster. The differences between the different NGS platforms lie mainly in the technical details of the sequencing reactions. They can be divided into serial sequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor sequencing.
The next generation sequencing provides ultra-high throughput, scalability, and speed. Currently, the next generation sequencing play a huge role in clinical research, reproduction health, environmental, and forensic science. The cost of sequencing for a complete human genome is now down to less than $1,000, allowing the entire human genome to be sequenced in less than 1 day. And now sequencing has entered a new phase, and it is truly an important milestone in the progress of recent bioscience research. Thanks for watching.